And you say, well, they might come on meteorites. Well, they don't. They don't come in an antimerically pure form on meteorites. They come as gross mixtures, and it would be very hard to use them.
I don't think meteorites make the right sort of mixtures.
For example, if you have a protein, and a protein has a 7-year half-life, if you have a single 200-mer polypeptide in water, if you have one molecule of that polypeptide in water, then its lifetime is 2,555 divided by 200, 13 days. That's all you have. So somehow, if that one polypeptide formed that happened to be the right sequence to do something, it's going to have to find all the substrate that it's going to act upon very quickly, within 13 days. If you want to have the RNA hypothesis, let's just take a 600-mer of an identical RNase. Say you had a mole of these. Let's be generous and say it had a half-life at room temperature of 100 days. Again, that's probably pretty generous. Actually, time is a real problem. But that's the problem. When the free energies are positive, these reactions favor, as you know, the starting material. These want to hydrolyze. And these are the types of times you have. Joshua Swamidas told me when we were discussing this.
You have to cross that bridge one time.
And you only need one RNA strand. That's all you need. Well, you got 4 hours then. It's going to have to find the substrates that it's going to act upon. Just to give a little bit of background, here's the 20 canonical amino acids. All the ones in red circles have these active side chains, and the ones in dashed circles are reasonably active. Now, Matthew Powner, you have aminonitriles.
Doing this in a prebiotic setting.
Paul Rimmer has suggested that your aminonitriles would work. They can't, because there is no aminonitrile. You have to start from these amino acids that we know, these are the starting materials that people have argued for many years could be made in Miller-Urey type chemistry. And also, if you start with your aminonitriles, you have no stereo control here. So starting with these two acids, get this product. Paul Rimmer tried to answer this question, and poor Paul, he started with a different material. He started with an aminonitrile. That's not what's given here. Plus, the aminonitrile would have never controlled stereochemistry, so everything was planar. You've got to show stereochemistry because that's going to be important for making polypeptides. Reza Ghaderi, how about your carbonyl sulfide chemistry, which is prebiotic? Will this work, or will it have side chain competition here? How about Bruce Lipschitz? Will your hydrophobic pockets work for this? Bring this forward and see if the three judges agree that you could cleanly get this dipeptide in greater than 90% yield. And condensation polymerizations generally need to be in 99.99% yield to give you any decent molecular weight compound. 90% yield is being very generous on this. This may be the easiest question among all the five. John Sutherland has said.
But we need to have more constrained chemistry to actually make the right sort of mixtures.
Nobody ever has made glucose in its enantiopure form from the foremost reaction. And nobody has ever even separated what they've been able to make because it's unusable. Foremost reaction doesn't provide it. Here's what nature has to go through to prepare glucose. Doesn't use the foremost reaction because that would be unusable, that material. Just to make glucose, it takes 11 different enzymes. 4 of those enzymes are unique to glucose formation, and it takes 4 activators on top of that. So there's 15 different activation steps, enzyme-induced and activator-induced. 4 of those enzymes being unique. And so you have all of these different enzymes. And you say, well, this enzyme might form randomly. Well, if this enzyme were to form randomly, it would be 10 to the 6,368 possibilities. That would take you more time than 10 of our universes to do this. So you know that this is not going to form randomly and then fold up random. And if you add up all the amino acids, all of these polypeptides, it's 10 to the 65,000 possible combinations here. You know this isn't going to happen. Here is the enzyme, phosphorylase. This is the enzyme that dimerizes glucose. Look at this enzyme. It's 98 kilodaltons. That's a molecular weight of 98,000. It has 842 amino acid units, 10 to the 1094 possible combinations. And then it has to get folded right and all of this. So this is what nature has to go through just to dimerize glucose. You guys think that we're really not far from cracking this problem. Some of you have even said that we're all set up now for Darwinian evolution to take over. Well, let's see. Can you even just make the dimer? Because you guys know that you're going to have to do this thousands upon thousands of times in a prebiotically relevant manner if you want to say that we started with the sugars. And you can't just say enzymes. I mean, this YouTuber just said, well, enzymes. Who of you is going to give an exam and just say, how would you carry this out? And somebody writes enzymes. Why don't you just write with chemicals? If you think that's an answer, what enzyme? What enzyme is going to do this? Is phosphorylase hanging around here? What enzyme are you talking about? Origin of specified information rather than Shannon information. Shannon information is trying to gather information out of random sequences. But specified information embedded in the sequence in polypeptides, polynucleotides, or polysaccharides, any one of the three, whichever you think arose first. And consider how that would translate its information in a relevant time if it's just one polymer molecule. Think about that. But the question is, what is the origin of the information? How do you gather together the information you're going to need to build a cell? I don't know. YouTubers have said it's already encoded in the DNA. No, that's Shannon information if there's no particular sequence to this thing. Origin of information. Critical for life is the origin of information, DNA and RNA, the order in which these things are attached. This information is primary, matter is secondary. So that information has been here, it's been on a piece of paper, it's been on SRAM, it's been on flash memory, now it's in an RF wave. Then when it hits that box, it goes down a wire. That information is going down a wire, then goes through a server farm into another flash memory. The matter upon which it resides is secondary. The information is primary. The information is the key. Nobody knows where this informational code came from. If somebody tells you that the DNA itself is the code, that's a bunch of garbage. That's like saying this memory stick. This is the information. I haven't written anything on it, but that's inherently the information. No, this is the medium upon which it's stored. Where did the blueprint come from? Where did this specified information come from? Assuming you had access to all the polypeptides that you wanted, your choice, whatever you want, all the enzymes you want, that's fine too. All the polynucleotides, DNA, all the RNA. And I'll even give it to you in any sequence you want. So you can have any sequence you want. I'll give you the information. It's yours. And you have all the polysaccharides, which are the hardest class to make, as you know. All the polysaccharides you want. And I'll give you all the lipids of your choice. Could you, in your research group, not on a mindless early Earth, even in your research group, in your laboratory, could you assemble those in your lab into an integrated, functional, living system, namely a cell? Could you assemble those into a cell? Even if you had all these molecules, even if you could make all these polymers, which I don't think you can because you can't solve numbers 1, 2, and 3. And even if you had the informational code, the answer to question number 4, how would you address in your laboratory, not trying to figure out how it did it on a mindless early Earth, how would you figure out in your laboratory to make the simplest of cells? And remember, a cell is going to have these characteristics: responsiveness to the environment, growth and change, ability to reproduce, have a metabolism and breathe, maintain homeostasis, and that's what constitutes a cell. And you have to be able to pass on traits to offspring. This is the characteristics of life, not my definition, textbook definition. I don't understand why there's this suggestion that we're very close to solving this problem on origin of life. But you have to solve all 5 of these questions in order to make life. I'm just asking you to solve one. These guys who work in origin of life are absolutely clueless. Everybody's clueless on this.